Hello and welcome to How Genomes Think. The topic today is going to be around genome dynamics and specifically how we can analogize that as thinking to make it very intuitive for us humans to understand how genomes behave in actual nature. And this includes, of course, cancer cells, because why do cancer cells actually kill you? Aren't they the same cells? Aren't they your cells? Why do they go rogue, and why do they have an interest, or perhaps not an interest in killing you, but a disinterest in having you survive? Why is that? Well, I can kind of spoil it a bit. It is because of genome dynamics, and it is precisely what we're going to talk about today. Before we get into that though, my name is Merlin. I've got a Bachelor of Science in Molecular Biotechnology. I'm one of the co-founders of Marchwood, where we help cancer patients outlive the disease. To that end, if you are a cancer patient and you want to socialize with others and with other cancer patients, that is, and want to outlive your disease, you can find our community project at marchwood.com/outlive, where we try to do just that, help you socialize with other cancer patients and help you outlive your disease. I hope to see you there. Let's get into the video. So it's going to be a bit of a different structure of a video. It's going to be more of a lecture kind of way, or just, me talking about the things I find interesting about this topic, and I hope that they're helpful. I hope that this video structure is something you like. So just, drop a comment if you like it, drop a like if you like it. Tell me what I did well, what I didn't do so well, and I'll hopefully improve. Now, I think it's important at the start to define terms, because when we asked at the start the question, why do cancer cells kill us if they are our cells? It's very important to at least understand what even makes a cell ours. Like, what differentiates, our cells from our brother's cells, or our cells from a lion's cell, etc. And you can do that at arbitrary genetic distance. But what makes a cell ours? And maybe what makes cancer cells different? Because apparently they don't behave like the rest of our cells. I mean, the rest of our cells are trying to keep us alive. Cancer cell maybe isn't actively trying to kill us, but it's certainly not necessarily actively trying to keep us alive. And why is that? That's a very important discussion. So, biologically speaking, what differentiates your cells from your brother's cells, from a lion cell, from a bacterium cell, the primary distinguishing factors, and there are others, of course, but the primary distinguishing factor is the genome. If the genome is vastly different, you are vastly different people, or even species. And that's kind of where the difference lies with cancer versus your own cells. So if we, for example, take here a normal healthy cell in your body with its normal healthy mitochondria, which are the energy producers and the quality control units basically, and we take comparatively another cancer cell with its small and dysfunctional mitochondria that don't really work as they should, don't really produce good energy, and certainly don't work in quality control anymore, otherwise the cancer cell would be dead. We have, in these cells, we have two genomes. We have, on one hand, in both of these cells, the mitochondrial genome, which both of these cells have, and in both of these cells, in the cancer cell and in the healthy cell, these mitochondrial genomes are, it's called conserved. So they are the same. They're monoclonal, basically, meaning that the mitochondria here and here are basically the same organism, or the same endosymbiotic organism, because they can't survive on their own. They can only survive within our cells, but they kind of act like their own organism because they have their own genome. Now, if we look at the nuclear genome, which is the second set of genome that a cell has, there's two things that differentiate them from mitochondrial genome. Firstly, the nuclear genome is vastly more complex than the mitochondrial genome. It's vastly larger, has vastly more genes, and is vastly more complex than the mitochondrial genome. But the nuclear genome is different in cancer cells than in normal cells. So if you take mitochondrial genomes and you would sequence this and this, you would see that mostly these are still like these. Now, there are some mutation or some damage that happens that doesn't get repaired because of the mitochondrial impairment, but that damage can be repaired if mitochondria are reactivated. And in nuclear genome, this is a different story because every cell has one or two nuclear genomes, depending on where in the cell cycle, so the cycle where the cell goes through replication and divides itself and kind of grows, depending on where in that cycle the cell is, you will have one or two nuclear genomes. You will have, however, 100 to 10,000 mitochondrial genomes. And you can kind of see here already why it may be easier to repair these genomes if they have a bit of damage than this genome if it has a bit of damage. Because with the mitochondrial genome, you have like 100 or 100 to 10,000 copies to kind of compare and to throw everything out that's, doesn't work and to leave you with healthy mitochondria once again. You can't really do that with the nuclear genome because you only have one genome per cell. So if this nuclear genome of the cancer cell has a mutation, you don't have a nuclear genome and a second nuclear genome to compare it to unless you're specifically in this part of the cell cycle. So there's a part of the cell cycle where the genome is replicated and then you have two. And if damage occurs during that, then yes, you can repair that. You can repair that with high fidelity. Now, the cell has some tricks where it will try to basically repair the damage because you have a paternal chromosome and a maternal chromosome, and it will try to kind of cross-reference paternal, maternal, and repair damage that way. So they're relatively stable, these nuclear genomes, but when damage occurs and damage propagates in both lines, you have no chance. You have no comparative thing like with the mitochondrial genome where you can actually compare and contrast and see what's wrong and what's correct. Now, the mitochondria can do that. And this is also the crux of the matter. This is why cancer cells aren't your cells. Your cancer cells have an entirely different genome. Now, both in structure, but also in how it's expressed, both in genetic sequences and regulatory sequences, the genetic makeup, the genomic makeup of the cancer cell, and this is the nuclear genomic makeup, not the mitochondrial genomic makeup, is different from your cell. So when you ask, why would my own cells kill you? Well, because genomically speaking, they're no longer your cells. Now, obviously, the distance between your cells and your cancer cells isn't as large as the distance between your cells and a lion's cell or your cells and a mouse's cell. So there is still, some similarity, but the nuclear genome is different. And that's why cancer cells just behave more or less like a bacterial infection and don't really care if they kill you. Because you're not them and they're not part of you anymore because they have a different genome. And here is where the helpfulness comes in of thinking that genomes think. Because if we postulate that genomes think or have a kind of innate drive, and they kind of do because genomes that don't have a drive, have this drive, they don't replicate. So if we presume that genomes try to replicate themselves, this is helpful because that's the behavior of genomes. Genomes try to replicate themselves. And if they don't have that drive, they die out. So in a very real sense, they kind of do think or want to procreate and to proliferate and to multiply themselves. And this is where, the kind of conjecture is very helpful, because if we conjecture that genomes actually have a mind or a will of their own, then we can intuitively understand them. Because if there's someone sitting across from you and he's not telling you everything, but you have kind of, you know how they are, you kind of know what their tendencies are, you know how they express emotion, you know kind of what they want in life, you can kind of understand what their psychology is and how they think and perhaps what they're not telling you. And the same is true here, because if you just presume that a genome thinks and that it is selfish and that it wants to multiply itself, you can intuitively understand why certain dynamics happen. For example, in cancer, why a cancer cell has no interest in you surviving and why it ultimately ends up killing you if you don't do anything against it. Now, there's a very, very, very good example of this in nature, where, for example, why does a lion kill the ape or the zebra or the gazelle and eat it? Why? I mean, that's also a genome. Why is it killing that other genome or stopping the proliferation and growth of that genome? Well, because it's not the same genome. So it has, again, this difference between one and the other. And here it's between a line, and I'm not going to draw a line. Maybe I'm going to write it. Lion and let's say that's a zebra. I can't draw a zebra and a lion, that's why I'm writing it. If you have the difference between a lion and a zebra, and the lion doesn't care about the zebra and kills and eats it because its genome is selfishly replicative or selfishly proliferative, then the same is true for the cancer cell and the rest of your body, which is kind of like a host for the cancer cell, much like you would be a host for a bacterial infection. And the bacteria and the cancer cell and the lion cares little about you as a host or the zebra as a food source, as each other. They just don't care. They want to proliferate. They want to survive themselves. They don't really care if they have to kill a zebra or if they have to kill you or if they have to infect you. They just want to survive. And this is also, by the way, a small aside, one of the problems that we're trying to figure out with sexual reproduction. Because in sexual reproduction, like if you take, first, I'm going to go back. If you take a bacterium splits in half, makes two bacteria, which have more or less the exact same genome. So this genome here is very happy because it's replicated in both of these. It's the same genome. Now, obviously, mutations do happen, and that's called genetic drift, and that's a mechanism of evolution. But largely speaking, the genome is propagated through the generations with relatively high fidelity. Now, if you look at sexual reproduction, you have a mother and a father who come together and give half of each their genes to the child. And then they should be happy that half of their genome is replicated versus here where the entire genome is replicated. Well, that doesn't make any sense. At least not just by, if you just look at the numbers here. Here's 100% or 99 point whatever percent replication, and here's only 50%. Why does that make sense? That's one of the big questions about sexual reproduction. There's some questions. Does it make significant improvements in survivability? Does it help against parasites? Because if you have a complex organism, a large body like us, you're prone to parasites or infections because you're just a growth niche, as much to other infecting organisms as to your cancer cells. You're a buffet, if you can avoid the immune system, it's very safe within your body. You don't have a lot of enemies, so to speak. All the other cells, apart from your immune cells, don't really attack invaders. So if you, for example, have a healthy cell just beside a cancer cell, the healthy cell doesn't really care, or it doesn't really understand that. It can't attack the cancer cell. Only the immune system does that. And that has a lot of reasons, because if your cells would attack each other, you would die pretty quickly. So it has sense that the immune system is very specific there. But this is a problem, right? This is a question. Why does this work? This is something we still have to figure out in genome dynamics. I would think it has something to do with mitochondria, but that is too deep of a water here. So how can we take this example of ecology of the lion and the zebra back to cancer? Well, again, the cancer has a very mutated nuclear genome. And it doesn't have two copies with which it can very nicely correct itself. And it doesn't have the ability to interchange nuclear genomes with other cells. So nuclear genomes between cells very rarely, if ever, transfer. And this is called horizontal gene transfer. It's a very technical term, just basically means a cell gives its genome or genomic material, genetic material to a different cell horizontally, not vertically as it happens down here. So this cancer cell is out of options. It is a different organism than you, so it has no incentive in letting you live. It can't really be brought back to the light by giving it a better, nuclear genome and healing it that way. I mean, perhaps technology advances and we can do that medically, but it doesn't happen in nature at least. And the mitochondria here, as I've stated at the start, they're dysfunctional. And one of the main functions of mitochondria is to kill defective cells. And if they are dysfunctional, if they just don't work, well, how will they kill the defective cell? They can't. So, that's kind of the whole point in cancer, is that your nuclear genome becomes vastly different than your normal cells, which is why cancer development takes years to develop, because it doesn't suffice to just have suppressed mitochondria. This happens all the time. If you eat a cake, you will have suppressed dysfunctional mitochondria. That's just how it happens. Not dysfunctional, you will have suppressed mitochondria. And if you have that suppression for long enough, you will have dysfunctional mitochondria, but you still won't have cancer. Why? Because you need this mutated nuclear genome alongside the dysfunctional mitochondria to have cancer. The cancer cell needs to develop and evolve into an entirely different organism for the genome dynamics of cancer to make sense. Okay, but the mitochondria are the same. I said that in the start. The mitochondrial genome is the same between these and these. More or less, I said there's some damage here, but we can repair that because we have so many mitochondrial genome copies per cell. And another cool fact, we actually see that there is horizontal gene transfer between mitochondrial genomes from healthy cells to cancer cells and vice versa. This is very cool. So for example, there's a study that shows a healthy macrophage, how healthy macrophages are manipulated by cancer cells, by the cancer cells shooting unhealthy mitochondria into this arriving macrophage. So I'm going to turn the page here, give me a sec. So if we have if we have a healthy macrophage here, and that's not how they look with healthy mitochondria, and this now migrates into a cancer tumor with multiple cancer cells, right, with their genetic material, their nuclear genome, and then their dysfunctional mitochondria. When it comes here and arrives here, these cells sometimes, and this is not, I don't know if this is always the case, but in this study, they actually found that these cancer cells will transfer dysfunctional mitochondria into this macrophage. Why is that bad? Well, if you get more and more dysfunctional mitochondria in here, and you know that mitochondria are highly dynamic, so mitochondria don't just stay segregated like this, they split up, they fuse together, and now you have this dysfunctional mitochondrial material that's coming into the healthy cell, and the mitochondria in flux, so they incorporate this dysfunctional material into the mitochondria here. That includes genomic damage to the mitochondrial genome. And now we also see that these cells, these immune cells, become affected negatively by these dysfunctional mitochondrial materials by, having them absorbed or having them ferried into themselves by the cancer cells. Now, there's also a bunch of other things that happens here that make these immune cells then inactive. One of the things is acidity. So cancer cells will, at least when they're growing, when they're proliferating, they will maintain a very highly acidic environment. And they do that if you postulate that they can think, they do that because this high acidity around them is kind of a shield for them. So this acidity does multiple things. If you have an incident immune cell like this macrophage, the acidity already hampers with its activity. It stops some of the signaling of the immune cell to the cancer cell. So there are immune cells that can actually tell cancer cells to kill themselves. This signaling doesn't work as well in the acidic environment. So that's a layer of protection for the cancer cells. Secondly, this acidic environment destroys neighboring cells. So it gives the cancer cell room to grow. Then it also destroys the extracellular matrix in some ways, which allows the cancer cells to decouple from growth control from that extracellular matrix, which is a structure that in complex organisms, in complex eukaryotes, such as us, as mammals, has very important functions in guiding and regulating cell growth. So it decouples them from that. Not entirely, depends on the cancer specifics, right? It's obviously very, very complex, but it does that. And then also the acidity kind of signals back and improves energy metabolism within the cancer cells themselves so that they can grow more quickly. Again, if we take this back to genome dynamics, of course, the nuclear genome here in the cancer cell has very much a reason to keep all of this up, to keep all of this damaged mitochondria up, because again, mitochondria here, whether they're destroyed or not, they have theoretically the capability, when they function well, and this is the mitochondrion, right? When they function well, they do good energy production, and they can, this should be a skull, right? They can kill cells. So mitochondria can kill cells, and they can do very efficient oxygen-dependent energy metabolism, but they don't allow this rapid growth of cancer cells, and they don't support this acidification. So obviously the nuclear genome has a high interest in keeping mitochondria suppressed, because this is a very big risk for cancer cells, and the mitochondria have a keen interest in killing the cancer cells because, and this is a trick again, if we go back to this, I already told you that the mitochondrial genome is very much conserved and monoclonal across your body. So the mitochondrial genome is the same across your entire body. More or less, but because of these gene copies, it's the same. This means that if you take these mitochondria and compare them between cells, they are, genomically speaking, the same organism. That's like comparing your finger with your eye. Yes, they're different, they're in different places, they have different functions perhaps, but they're the same organism. They're all part of you. And it's the same here because they have the same genome. They're basically the same endosymbiotic organism or network of the same monoclonal organism throughout your cells in your entire body. And as I've said, they fuse and split up within cells. And I already told you that they have horizontal gene transfer between cells. So there is a very vibrant, dynamic network between all of our cells that kind of have a very, very stable genome, especially compared to the nucleus genome, which is way more complex and way less stable. And that means, or it's perhaps not more stable, but it is less complex, and it is in far higher copy number within our body, so we can repair any damage that occurs to the mitochondrial genome. So where am I going here with? We have the nuclear genome of the cancer cell. So we have the cancer nucleus and its interest, right? Cancer nucleus. This has its own interest. This is selfishly proliferative. And since this is unequal to your healthy nucleus, it has no interest in the proliferation of your healthy nucleus. And that's bad for you because you're defined by your healthy nucleus. All the cells that you call your cells have this healthy nucleus. So if the cancer cell has no interest in the proliferation of your healthy nucleus because it genomically is a different organism because of the accrued mutations, then we run into a problem. Here's the nice thing though. We also have mitochondria. It's difficult to write here. Mitochondria. And they have their own genome. And this genome is both inside these and inside the cancer cells. So this genome, because it is here, and it's much vast, like because your healthy cells outcompete your cancer cells in number, 1,000,000, 1,000 to 1, whatever it is. They're far more healthy cells than cancer cells, and the mitochondria in your healthy cells are happier. They have a vested interest in having these survive over these. But they are inside the cancer cell. So they can influence the cancer cell theoretically. And that's where this kind of comes in. Because they have an interest in our survival, we can use them as kind of sleeper agents within these that we perhaps can activate. And that's a ZZZ sleeper agent, right? So if we can activate mitochondria and they have an interest in cancer cells not proliferating, not killing you, they don't necessarily have a direct interest in killing cancer cells. They just have an interest in them not killing you, right? So they're kind of, you could say, either sleeper agent network, or perhaps they're diplomats between your cancer cells and your healthy cells. And if your cancer cells misbehave and threaten your healthy cells, the mitochondria will try to kill the cancer cells. The other thing that happens if we reactivate mitochondria is that we get rid of this acidity. This acidity happens because primarily in the first step of tumor genesis or cancerogenesis, this happens because of mitochondrial dysfunction, which leads the cell to the budding or developing cancer cell to use lactic acid fermentation, and this lactic acid fermentation, as you can see in the name, has a lactic acid molecule as the end product. And that is excreted as waste from the cancer cell into the environment. And this is where this acidity comes from. So if we can reactivate mitochondria in these cancer cells and make them healthy, happy mitochondria that work well, we can get rid of this acidity, or at least we can get rid of the production of new acidity. There's also ways of directly pharmacologically stopping this or neutralizing this acidity or helping your body neutralize it. But we're talking about genome dynamics, so we care about mitochondria in the first place. I'm going to get into neutralization of that acidity in just a bit. So, mitochondria, rehabilitation. We want them to be powerful enough to kill cancer cells. How do we do that? How do we go from a dysfunctional network in the cancer cell of small, fragmented, not very noteworthy, dysfunctional, incapable, unpowerful mitochondria to nice and active, big dick energy mitochondria? I shouldn't say that, but active mitochondria who have the power to actually initiate apoptosis in cancer cells, which obviously something cancer cells don't want, because they just want to grow, grow, grow. But if we can get mitochondria to this point and they have the capability to kill cancer cells, one thing is guaranteed to happen. Now, this is very difficult. This is very questionable. Is this possible? Seems so in the literature, but how far this process can be done, is up for debate and will be shown in the future. But it seems preliminary results that this is possible, and that's kind of the whole point of our mosaic method, is to do this. But if we can activate mitochondria to have this capability, they can give the cancer cells a choice. They can say to the cancer cell, and this is again anthropomorphizing them, making an analogy how genomes think, so we can understand it more intuitively. They will say to the cancer cell, hey, either behave, right, and stop killing our brothers, and their brothers are the mitochondria and all the other cells, or we're going to kill you. And they will go into suicide for the other mitochondria because, remember, they have the same genome. So they're not really killing themselves because the genome lives on. If you have a clone, are you really killing yourself? Genomically speaking, no. Psychically speaking, religiously speaking, whatever. Doesn't really matter for the discussion. Genomically speaking, no, you're not killing yourself. If there's 2 genomes of you, you're just, deleting one genome. You're deleting one individual, but genomically speaking, the genome is still there, so you haven't killed yourself. Genomically speaking, this is very important because this is a difference. So if they kill themselves, genomically speaking, they haven't killed themselves because they're all our other cells. So this is a very real threat to the cancer cell. This is what we want. If we can give cancer cells this choice, behave or die, what do you think cancer cells will do? Well, they will either behave or they will die, right? So what tends to happen when we do see this in patients, in literature and elsewhere, is that cancer tumors shrink, below detectable thresholds. At least they stop growing, and they stop being able to kill the patients because only a growing tumor kills. If your tumor as it is now doesn't grow any further, you don't die because you're still alive. You're watching this video, you're up and well, but maybe not well, but you're up, you're alive. So if mitochondria can stop cancer from growing, which they can with this choice, right? With giving cancer cells this choice, they can stop cancer cells from growing. Then you can, theoretically just outlive cancer. And because mitochondria, again, they work to stop the accumulation of acid in the tumor environment. And because they stop this, they actually enable anti-cancer immunity to run more smoothly. Now, that's not a guarantee that it will work against cancer, right? But they at least help that effort. And there's also other things like neutralizing that acidity directly that can help, and we'll get to that in just a bit. But this is the major step. How do we go from these dysfunctional mitochondria up here to these nice and healthy and powerful mitochondria that can give cancer cells this choice? There's a few things. I think firstly we need to understand what's the difference between these, or what are the differences between these and these. Now, one of the things is that these are in a state of what is known as mitochondrial hyperfission. Hyperfission just means that they are fragmented. They are overly fragmented. That's what this means. One of the things that leads to hyperfission is actually, wouldn't you know it, sugar. This is a kind of a drawing of a sugar molecule, a schematic drawing. There's some hydroxyl groups missing, but the point is, this is sugar. If you consume a lot of sugar, this leads to the suppression of mitochondrial metabolism because sugar is not necessarily, or the first step in sugar metabolism is never made in the mitochondria. So sugar metabolism has two steps. First, into two smaller fragments, and these smaller fragments are then put into the mitochondrial metabolism and burnt there. This first step needs no mitochondria. And because this first step doesn't need mitochondria, if you use a lot of sugar as energy, you will see that your mitochondrial activity decreases because no longer is all your energy production pathway going on in the mitochondria. So if you, for example, take fat, let's take fat here, a nice long fatty acid, that needs to be metabolized in the mitochondria. It goes through various pathways of metabolism, and all of these happen in the mitochondria. So you can see why this would be more activating and kind of leading to higher activation of mitochondria than this. But this still does not explain hyperfission. So why does this happen? Well, if you have this glycolysis upregulation, for whatever reason, just simply having glycolysis active leads to more fission in mitochondria. There's some signaling that happens. Maybe it's because of lowered oxidative stress, which is kind of counterintuitively, but the mitochondria will split up. And when they burn fat, and this fat burning actually causes a lot of reactive oxygen species to be produced, then they will fuse to be more efficient and to kind of buffer that production better. And when they fuse, they will also produce something known as glutathione, which protects against reactive oxygen species. So this is kind of a self-protective reaction of the mitochondria to fuse more together to bigger mitochondria, to bigger conglomerates that are more efficient at energy production. And this is kind of because they're not really activated, they just like kind of fizzle out and go into hyperfission and don't really do much, but like kind of burn this little thing, which doesn't require that much mitochondrial activation. And another thing that happens is that this pathway, in comparison to this pathway, depletes glutathione. And glutathione is our protection also of our genome from glycative, oxidative, and toxic damage. So having this state of hyperfission also leads to higher mutation in the mitochondrial, in the nuclear genome. This step, okay, one of the things we know, stop sugar, use fat, because that promotes this, right? One of the things that also happens in this, which is why this works, so if we go from a sugar metabolism, which, depletes glutathione, to a fat metabolism, which repletes glutathione, parallelly, this step here, this down here, produces a lot of a molecule known as NADPH. This NADPH enters also mitochondria. It's a balance between the rest of the cell and the mitochondria, but it also affects mitochondria in the way that the mitochondrial molecule known as cytochrome C, it's bad writing, sorry. This cytochrome C gets put in a, how do you say, it's not an inactive state per se, but it's put in a state by this metabolism, by this NADPH, that prohibits it from doing this. And that's of course bad, because if it can't do this, it can't give cancer cells the choice, cancer genomes the choice of behave or die. And all of this, what we're trying to do, kind of goes out the window. So, step one, don't eat sugar. And this includes carbohydrates. So, I think I need to explain this as well. If you take, like a loaf of bread, that's a very pretty loaf of bread. This loaf of bread is directly digested into sugar. It's not digested into carbohydrates, it's comprised of carbohydrates, and these carbohydrates are digested into sugar. And this sugar then fuels all of this nonsense, which we don't want happening. This fuel leads to higher NADPH production, which leads to cytochrome, to a shift in cytochrome state away from the state that cytochrome needs to initiate this apoptosis, to initiate the death of cancer cells, also of normal cells if they are defective, but also cancer cells in this example. And it leads to the upregulation of glycolysis, which leads to hyperfission. And there's even a bit, even worse thing that happens. If we are in hyperfission, I'm just going to pile this on. If we are in hyperfission, what doesn't happen is that these mitochondria don't come together anymore. When they're in hyperfission, they don't fuse a lot, which is kind of in the term. So they don't come together and make these big conglomerates if you're eating a lot of sugar and putting them in hyperfission. And that is bad because now you have all of these mitochondria that aren't very active, and you have depleted glutathione, right? Glutathione, which remember is kind of the protection of our genomes and of our cells entirely against toxic oxidative and glycative damage. If you're in hyperfission or if you're in glucose metabolism, this depletes. So this goes down, which is bad because this protects our genomes, both our mitochondrial and our nuclear genome in this cancer cell, from more genomic damage. So what's happening in this hyperfission state is that they don't fuse. This means they can't exchange materials. Mitochondria need to fuse to exchange their genomes and their, shall I say, machinery. They need fusion for this to be exchanged. And we want this to be exchanged to sort out damaged machinery and damaged genomes and multiply the working machinery and working genomes, which happens in this state. And then these nice large mitochondria can sort the destroyed genomes over here and the happy genomes and machinery over here. And then they can split. And this is destroyed and recycled, and this lives on. Hyperfission, meaning glucose metabolism, glycolysis, just destroys all of this. Sugar and carbohydrates, so bread, rice, potatoes, cakes, sweets, you name it, anything that has carbohydrates in it, bad because of this. Now, there's obviously a difference in different carbohydrates, how bad they are, etc. But generally speaking, if you're a cancer patient, you should immediately stop consuming carbohydrates and you should replace it with fat, because studies have shown that you simply have better survival chances if you're in ketosis versus not in ketosis. And ketosis is the state of burning and using fat for energy. So that's kind of how genome dynamics works in cancer. And I'm going to summarize everything we talked about today. So in cancer, we have a cancer cell. And this cancer cell has a nucleus. And it has a bunch of dysfunctional mitochondria. Now, this cancer nucleus is a different organism from the cancer nucleus is completely a different genome, or carries a completely different genome than the rest of your cells. And this makes the cancer cell a different organism to the rest of your cells. It's unequal to the rest of your cells. The rest of all your cells, your specialized cells, whatever. And it has dysfunctional mitochondria. Again, why is this bad? Because mitochondria, when active, nice, big and active, can give a cell a choice of behave or die. These dysfunctional mitochondria cannot. These dysfunctional mitochondria are kept that way through hyperfission. And hyperfission is caused by sugar metabolism. And it is resolved by stopping sugar and adding more fats. That's it, basically. That's it. So we want to reactivate mitochondria. So when they become reactivated, they can give cancer this choice. Cancer will then either die or behave, i.e., not grow as abnormally rapidly and deadly for the patient. And these mitochondria will produce energy without producing acid. And that here is good because it allows the immune system to function properly. And then we have the holy grail of macrophages with their nice mitochondria coming in here and being able to kill the cancer cell because we rehabilitated mitochondria. Now, obviously, this is only a part of the mosaic method. This is is maybe a fifth of the formulated mosaic method, which you can find on marchworth.com/mosaic, by the way. But this is very important. This is the very basis of the mosaic method. This is the logic behind it. If we rehabilitate mitochondria, we can stop cancer. It's that simple. Now, the difficult part is, how do we do this, right? I started with some here, glucose, stop carbohydrate consumption, add fat, be in ketosis, stop this hyperphagia. But there's also some other aspects of nutrition and some other helps that we can give the body to do this and do this killing of cancer cells. I think that does it for this video because I think it's been pretty long. I don't know if you like this structure even of the video. You can tell me in the comments whether this has been good, because I think this gives me a bit more space to just, let my expertise shine, let me talk about the things I love talking about. And then you can ask questions in the comments. I will then make a video to the questions you make or the questions you pose and hopefully answer them that way. I think that was it for today. Yeah, this is the logic behind the mosaic method. Again, you can look at it on the marchworth.com/mosaic. And if you are a cancer patient and you're wanting to connect with other cancer patients and you're trying to actively outlive your disease, and you don't have anything against, perhaps a bit of a strenuous habit change, do check us out. Do check us out at Marchworth. I'm going to write that out. marchworth.com/outlive, because this is what we try to do for you. Outlive. With all that said, leave a comment, leave a like, leave a dislike if you hated it, leave a subscribe if you want to see more of this. Ask your questions, show me what I did wrong, what I did well. This has been the first video in this structure, and I probably have a huge room for improvement. Check out our website, check out the community project with which we are trying to help you if you are a cancer patient. If not, no need. Just enjoy the video, so don't. Let me know whether you are a cancer patient or not. I wish you swift healing from all the ailments you face and lasting health after that healing. May God bless you and your family with a long and most of all, happy and fruitful life. God bless.